The data presented in our ASCO abstract, Enrichment of Circulating Tumor DNA from Cell-Free DNA of Hematopoietic origin, show how we developed and validated the Nu.Q Capture methodology to enrich tumor-derived nucleosomes. The technology is potentially the basis of future cancer diagnostic tests. But here's some background first. Chromosomes from healthy cells are different from cancer cells, both in their genetic sequence, but also in terms of their protein structure due to epigenetic differences. When cells die, the chromosomes are fragmented by enzymes releasing their component parts, disc-like structures called nucleosomes. In fact, the majority of DNA in circulation exists as nucleosomes, where the DNA is wrapped around discs of protein like a cotton reel. The protein structures protect and stabilize the DNA, but they also retain the genetic and epigenetic information from the chromosome. Some of those nucleosomes contain linker DNA, which links them together within the chromosome. And a key difference between background healthy nucleosomes and the nucleosomes from tumors is that the DNA in healthy nucleosomes is slightly longer. It retains its linker DNA, whereas nucleosomes from cancer don't. Nu.Q Capture is designed to pull out and deplete these background healthy nucleosomes that still contain linker DNA, and so enrich cancer-derived nucleosomes which contain tumor-specific mutations.
We take clinical plasma samples containing nucleosomes with both shorter tumor-derived DNA and longer background DNA, and add magnetic beads coated with a protein that binds to the linker DNA. Only the nucleosomes with the linker DNA bind to the beads, whilst the shorter nucleosomes remain in the plasma. The magnetic beads and the bound larger nucleosomes are pulled down, allowing the shorter nucleosomes to be removed, and the longer nucleosomes can be recovered after the beads are released.
Once you separate out the nucleosomes and their associated DNA on the basis of epigenetic content, interesting things start to appear in the analysis. It's a bit like trying to listen to two songs at the same time. They drown each other out, particularly if one is louder than the other. By separating them out, it's much easier to hear the tunes. So once we've captured the nucleosomes, we have an opportunity to analyze them in a number of different ways. We can use mass spectrometry to help identify new biomarker targets, and we can sequence the DNA, which is now enriched for the tumor-derived DNA, and hope to use that for diagnostic applications, for treatment selection, and for disease monitoring. And finally, we think because we're now focusing on the tumor-derived nucleosomes, that will really enhance the sensitivity of our Nu.Q immunoassays for diagnosing different types of cancer using our nucleosomics platform. The data presented in our ASCO abstract, Enrichment of Circulating Tumor DNA from Cell-Free DNA of Hematopoietic origin, shows how we developed and validated the Nu.Q Capture methodology by first optimizing the protocols on nucleosomes from cells grown in the lab. We can clearly show efficient and reproducible capture, and therefore depletion, of the longer nucleosomes with linker DNA from the shorter ones with no linker DNA. We can quantify that depletion using our standard Nu.Q immunoassays targeting the histone 3.1 variants. And we also demonstrate beautiful separation by analysis of the DNA extracted from each fraction with clearly distinguishable peaks at 147 base pairs for the unbound nucleosomes and 167 base pairs with linker DNA for the bound nucleosomes. We then applied the method to 5 clinical colorectal cancer samples and 3 healthy control samples. Again, the data shows a clear separation of the peaks in the 2 fractions, and importantly, almost no change in the healthy samples, demonstrating a clear difference between cancer-derived and healthy nucleosomes. In conclusion, while further research is required, Nu.Q Capture has the potential to offer a simple way to enrich cancer-derived nucleosomes from cell-free nucleosomes of non-cancer origin, and therefore enrich tumor-derived cell-free DNA. And this will enable significant opportunities for the development of new diagnostic tools.